Hello and welcome to Miss Carol's Kitchen, which is now our new makeshift lab. So what we're going to be doing this week is we are going to be looking at transport into a cell and out of a cell. And the cell we're going to use is a plain old chicken egg, which could be considered to be one of the largest cells on Earth. There's some debate about that. But if you think about this, this is a single cell, and it's a little bit like a plant cell right now because it has something hard on the outside, but it's not a cell wall, it's a shell. And if I crack the shell, then we can see what's inside the egg, and you're probably pretty familiar with this anyway. And I don't have lab glassware, so I'm using a champagne flute for this, just so you can see the different stuff inside the cell. So of course we have the yolk in the middle, and then we have the egg white. Well, it turns white once you cook it on the outside. And don't think about this as a nucleus and cytoplasm. That's not how it works. Yes, the egg white, the clear stuff, has cytoplasm in it, but it's thicker than cytoplasm or cytosol would be because it's mostly protein. So this is something that's going to feed the embryo of the egg if there is an embryo in there. And that protein is called albumin. And so we have a lot of albumin there. And we do have the egg yolk, which has a lot of stuff that would feed a growing embryo if there was one there. But it's called the yolk sac. And all mammals actually have this as well, reptiles, and bird eggs, and an embryo would grow on top of that, and it would provide the nutrients. So if you're wondering about this little sort of white thing that you're seeing there, and you always wondered, oh God, am I eating a chick embryo when I eat an egg? No, there should be two of them there, one on either side of the yolk, and those are just specialized proteins that actually anchor the yolk into the middle of the egg. So they're attached at the sort of top of the egg and the bottom of the egg, and that just keeps the yolk in the middle where it's nice and safe, so it's not against a hard outside. However, what you didn't see is when I broke the shell here, there is actually this little membrane on the inside of the shell, and I'm peeling it away now so that you can see. And it's actually two membranes, if we want to be honest here. There's an outer membrane and an inner membrane. And you may have actually seen this membrane before if you've tried to peel a hard-boiled egg, and there's that little film there. That's the membrane. And so what we want to do is we want to do a little bit of a demonstration with this egg without the shell, but with this. So what we want to do with our egg is we just want to get rid of the shell. And an excellent way to do that is to use vinegar. This is backwards, but it's vinegar. And so you probably know that vinegar is acetic acid, and you may or may not know that an egg shell is made of calcium carbonate. And so basically what we have here is when this is dissolved in water, and this has some water in it because it's only like, I think 5% acetic acid and the rest is water, this becomes basic. So what we have is a base and we have an acid. And in grade 10 science, of course, you learned that that's a neutralization reaction. And so what's going to happen is the eggshell will dissolve. All right. And so what we're not really, I guess we're dissolving it, but we're really actually performing a chemical neutralization reaction here. And so that's going to get rid of our shell. And then what we're going to have left is an egg surrounded by a soft membrane, and the membrane is going to be semi-permeable. So you learned last week that a cell membrane is semi-permeable, which means that some things can come in, some things can come out, but some things are going to be blocked. What things? That's what we're going to look at, okay? One other note, if you do want to know where the nucleus of this is, there is something called the blastodisc. And it's located somewhere on the yolk. I can't see it on this particular egg, but it's just a tiny little area, and that's where the nucleus would be. So don't think of the yolk as the nucleus, but the nucleus would be located in a tiny, tiny area somewhere inside the yolk, okay? So today, what we're gonna do is I'm gonna do 2 eggs because I've done this many times before, and the sad truth is sometimes you get an egg to break. So I wanna do 2 just in case one breaks. And I wanna get some measurements. because what we're going to be doing is looking at transport into the cell and out of the cell. So think about this. If material is being transported into the cell, is the cell going to get bigger or is the cell going to get smaller? Not going to give that away, but I just want you to think about it. And for now, what I want to do is I just want to measure the egg. I'm just going to angle my camera down so you can see what I'm doing here. Oh, and I have a plant in the way. That's really fun. Okay. So what I'm doing is I'm just putting the egg on a piece of paper, and I'm just going to make a little pencil mark here at the top and make a pencil mark here at the bottom. One at the side. I'm trying to hold my pencil straight up and down. There. And I'm going to measure that, and we're going to call this egg number 1. I will be careful to keep these straight. I'm going to do the same for egg number 2. P.S. These are Mr. Cooper's eggs, which he dropped off the last time he was in Sackville. Okay, and so that's egg number 2 right there. Okay, so what you can see is some pencil marks, and I'm going to go ahead and measure those. There's my plant again, got to get that out of the way. Okay, I'll fill those measurements in after, and for now, what I have is no beakers, but I do have plain glassware. This is filled with vinegar. So this is egg number 1. That's going to go in the vinegar, and we're going to see what that looks like tomorrow. Okay, if you go on the video chat today at 11 a.m., I'll do a quick check-in just to see what they've done in 2 hours, and then we'll check finally tomorrow. And the second egg goes in here. Okay, so what we have is 2 eggs, each in a glass full of vinegar. Oh, and you know what? You can see some stuff happening already. I'm not sure if you can see this, but you can see the bubbles forming. So this would be a little bit like in Science 10 when you had your Tums and your acid and you saw some bubbling happening. Okay, so what that is the release of carbon dioxide, because calcium carbonate contains oxygen, and that's going to turn into some carbon dioxide, which is being released. Okay. Now, once the shell is gone, what we're going to do tomorrow is we are going to put it in some distilled water. So you can take a look online and see what distilled water is if you don't already know. And then the next day, we're going to put it in some corn syrup, which is, let's see the ingredients here. Glucose, fructose, water, molasses, and salt. We'll talk about that later, okay? Stay tuned. Thanks for watching.